Cell cycle. What do you mean by cell cycle? A cell grows and divides. Before division, a cell undergoes following changes. What are the changes? The changes are first change in the shape of the cell, then volume of the cell, and then there is series of events which lead to change in first one surface to volume ratio, then second one size of the cell, that is decrease in surface area. Now the events are organized into three phases. One is G1, the second one S phase, and third one is G2, and another one phase it is M phase. The period between two subsequent cell divisions is called as interphase. This interphase has following stages. That one, first one is G1, which is also known as first gap period. Then second one, S phase for synthetic phase, and then third one, the G2 phase, which is for second gap period. In a 24-hour cycle, we can see that a cell rests about 10 hours in G1, 9 hours in S phase, and 4 hours in G2 phase, and 1 hour in M phase. This G1 first gap period, it is a major gap period in the cell cycle. It takes about 10 hours, or it is the most abundant time period taking phase. Here following changes occur. The first change is synthesis of mitochondria and ATP. Synthesis of mitochondria and ATP. The second one, change in the ribosome number. Third one, synthesis of RNA. Are the major changes which takes place during G1 phase. Then during this S phase, synthesis of DNA. S phase is characterized by synthesis of DNA. Then synthesis of histone proteins. Sometimes the question asks in PMD where division of centriole takes place. This is centriole division takes place during this S phase, and the DNA value doubles in this phase. Suppose a cell contains 2C amount of DNA, then after S phase, the amount of DNA will be 4C, which is also known as C value paradox. Then after S phase, the cell enters G2 phase, where duplication of all other cell organelles takes place. Duplication of other cell organelles. Then second event is synthesis of microtubules, which is also another one major event required for cell division. Now during this M phase, which is about 1 hour, the cell undergoes karyokinesis followed by cytokinesis. And what are the major events of karyokinesis? Karyokinesis means carrier, which means it is nucleus. Then another one, kinesis, it means division. So karyokinesis means it is the nuclear division. It takes place in 4 phases. The first one is prophase. The second one is metaphase. Third one is anaphase, and the last one is telophase. Then what are the major events of these? Now in prophase, a cell loses its nuclear membrane, chromatin material condenses, it forms chromosome. This one it is degrading nuclear membrane. This one it is chromatin material. Now centrosome divides to form two and they start migrating towards opposite poles. These are the major events of prophase. Then second one is metaphase. During this metaphase, the nuclear membrane is absent. The chromosomes are maximum condensed. Each chromosome has two chromatins. They arrange at a particular line at the equatorial plate. And the spindle fiber from opposite poles attaches with the centromere region of the chromosomes. Each chromosome has two chromatids in this phase. Each chromosome has two chromatids. This one is centromere. These are the spindle fibers. This spindle fiber formation is blocked by some chemicals like colchicine, vinblastine, vincrustin, which are useful in plant breeding process. Sometimes the question asks, which of the following chemical help in blocking the spindle fiber formation? These are the spindle fiber. It consists of microtubules, and this colchicine block the polymerization of microtubules so that the spindle fiber will be formed. So the chromosome number becomes double during this stage if they are connected with colchicine, which helps in formation of polyploid. These are the question which usually comes in DMT, that which of the following chemicals, and in that question the option contains either of these chemicals. Now another one phase is anaphase. This anaphase, if you will see here, the spindle fiber contract on both the sides or from both the poles. So a pressure is created on this centromere point, due to which the centromere splits in the middle. Due to splitting of the centromere, now each chromosome becomes single chromatid in nature, and after that again further condensation or contraction of spindle fiber takes place. So chromosomes start migrating from the equator towards the poles. Now in the anaphase, what we can see? In anaphase we can observe that the migrating chromosomes are showing different shapes such as V, L, J or I shape, basing on the position of the centromere. If the centromere position is at the middle, then it will show V shape. If centromere is towards one side, then it will show J shape. And if it is at one end, then it will show I shape. Now these are the migrating chromosomes. Now in this phase, there are three pairs of three chromosomes. Now you can see that there are three chromosomes migrating towards upper pole and three chromosomes migrating towards lower pole. But the difference is, previously each chromosome during metaphase has two chromatids. Now each chromosome has one chromatid. Now it's the last phase, which is telophase. This telophase is just the reverse of prophase. In prophase, what happens? Nuclear membrane disorganizes, spindle fiber formation begins, contraction of chromatin material takes place. But during this phase, what will happen? The chromatin or the chromosomes, now they will decondense. Nuclear membrane and a nucleus reappears. Spindle will dissociate. Now at the end of this phase, so what we can observe, that each cell has two nuclei and each nucleus is having 2n number chromosomes as that of the parent cell. Now it ends the telophase. After that, cytokinesis takes place. During this cytokinesis, division of cytoplasm is seen, and this takes place in two different ways. In case of plants, it takes place by cell plate formation method, and in case of animals, it takes place by cell furrow formation method. In case of animals, what happens? First, in the cell there are two nuclei, which is the result of karyokinesis. Then a contraction or a contractile ring is formed at the middle, which contract gradually so that the cell becomes dumbbell shape, and finally it touches at the middle point so that two spherical cells will be resulted. But in case of plant cell, a phragmoplast originates in the center, which is formed by endoplasmic reticulum, Golgi complex, and this phragmoplast grows in both the directions to touch the cell wall and finally separate the two cells. Now the upper diagram is for animal cell and the lower diagram is for plant cell. Now at the end of mitosis, two diploid cells are resulted, which are having same number of chromosomes as that of the parent cell. A cell divides in every minute. A cylinder contains some cells which occupy 2 cm height at 9 am. Now there are two questions attached to this one. First one, how much minute will be taken for the cells to fill up 8 cm height of the cylinder? The second question, if there are 32 into 10 to the 5 cells at 9 am, what will be the number of cells at 3 am? Now the answer to the first question. What is this one? This height is how much centimeter? 2 cm. Now in this question, what is the height? It is 8 cm. Now the cell divides in every 1 minute. Means suppose there is one cell in the cylinder. Now after 1 minute, what will be the number of cells? A cell divides in every minute means first one is 1, then the second one will be 2 cells, because it will divide, it will become 2, and this time period is 1 minute. And after second minute, the total number of cells will be 4, and after third minute, the total number of cells will be how much? It will be 8. Now in the question, it is given that the cylinder is occupied by 2 cm. Now after 1 minute it will be 4 cm, and after 2 minutes it will be 8 cm. So answer will be which one? This one at 9, 0 to 8. Now what is the formula behind it? The number of division is 2 to the power n, where n is this one, it is time period or number of times the cell divides. So 2 to the power 3, what is this one? 2 to the power 3 means it is 8 cm, 2 to the power 3 means it is 8. Now my question is that it is occupied by 2 cm. So after 1 minute it will be 4, and after 2 minutes it will be 8. So the first question's answer will be at 9, 0 to 8. The second question, the cell or the cylinder contains 32 into 10 to the 5 cells at 9 am. What will be the number of cells at 903 am? Now, how many divisions it will undergo within 3 minutes? 3 times division. It means the total number of cells will be 2 to the power n. This n is replaced by 3 here. So 2 to the power 3 cells. If there is one cell in the medium, then it will show only 2 to the power 3 cells. Now the medium contains or initial number of cells is 32 into 10 to the 5. So after 3 minutes, the total number of cells will be 2 to the power 3 into 32 into 10 to the 5. So this question's answer is option C. Now what the children do usually in this question? They see that there are this one 32 and here 65 is given, 120 is given, and they search for these numbers, not these questions which one containing this numeric as the power questions. And sometimes they think that this one is not correct because 64 will show only in 2 times division and 128 in 3 times division. But the answer is 2 to the power 3 into 32 into 10 to the power 5. And it is up only in 2013.